My name is Sherlyn Chen from Boehringer Ingelheim, and let me quickly introduce our 4 speakers. The first speaker is Dr. Neil Miller. He's from GSK. Neil is a scientific director within the System Modeling and Translational Biology group at the GSK. He's currently the team leader for a group of PBPK modeling specialists. Over the last decade, he has focused on the application of PBPK modeling in drug discovery and development, and is leading the vision to have a PBPK models in place for all compounds prior to in vivo studies in a model first, the experiment later paradigm. Our second speaker, Dr. Neil Parrott. He's from Roche Pharmaceuticals. Neil is a distinguished scientist in the Translational Modeling and Simulation Group, Roche Pharma Research and Early Development in Roche Innovation Center at Basel, Switzerland. Neil specializes in PBPK modeling and its application to Roche projects from early discovery to the market. Neil has a particular interest in the application of PBPK modeling for first-in-human PK prediction and to guide formulation and the pediatric drug development. He's involved in research activities to develop better PBPK models and is also active in cross-industry consortia. Neil has published over 50 papers and has given numerous presentations at international conferences. Our third speaker is Dr. Aki Heikkinen. He's the head of drug interactions and the transporters group at Atmoscope, Oulun, Finland. Atmoscope is a CRO focused on preclinical ADME talks. Aki is responsible for PBPK modeling as well as in vitro drug-drug interaction and transporter studies. His research interests are focused on translation of in vitro data into predictions of in vivo pharmacokinetics. Aki has published over 25 papers on PBPK drug absorption, transport, and metabolism. Last but not least, the speaker, Dr. Vera Lukchua. She's the director of simulation sciences at Simulations Plus. Vera has spent over a decade contributing to the development of GastroPlus software, and in her current role, she's supporting the teams responsible for the development of GastroPlus software and for modeling and simulation studies, helping companies in all stages of drug development processes. This webinar will be recorded and then will be available to the user group members in the future. Following each presentation, there will be an opportunity to ask questions. You can either send your question using the questions pane on your control panel, or you may ask your question directly using the hand raising icon. If you are using a telephone to listen to the call, please be sure to enter the unique audio pane displayed when you join the call. So this enables us to unmute your line so we can hear you. We now have a quick poll, and you probably see it already on the screen. So without further ado, we now begin the webinar presentation.
Thank you, Sherlyn, for the introduction. So good morning, good afternoon, and good evening to everyone, depending on where and when, if this is the recording you're listening to us. Firstly, we'd like to thank the GastroPlus User Group for this opportunity to talk about our publication, which was a very enjoyable and productive pre-competitive collaboration. Now, I think it's fair to say that there is scientific debate on the value of preclinical verification in PBPK modeling, and thoughts and opinions that I have heard include it's useful for passive distribution, so not when transporters are involved. It can lead and inform, but not verify. Animal empirical adjustments may or may not work for humans, and it also helps to explore mechanisms you cannot reliably predict from in vitro data, such as in vivo precipitation. So our hypothesis is that PBPK modeling with verification of predictive performance first performed in preclinical species is superior to empirical methods for predicting pharmacokinetics. And later on, we will ask you for your help in testing this hypothesis. Can we move to the next slide, please? To be of any value, hypothesis testing must have a firm foundation. And we will describe a strategy based on our considerable collective experience. This strategy, broken down into decision trees or flow diagrams, needs to be consistently and widely applied by you, our PBPK colleagues, to fully test the hypothesis. Can I have the next slide, please? Our strategy is based on the seminal paper from Hannah Jones et al. In 2006, titled A Novel Strategy for Physiologically Based Predictions of Human Pharmacokinetics, which, when I checked a few weeks ago, had been cited nearly 300 times. We updated the strategy of Jones et al. To include new knowledge and additional flow diagrams for each essential component of a first-in-human prediction. These updates were based on a comprehensive review of publications since 2006, and on the combined knowledge and experience of the group. Although we are all members of the GastroPlus User Group, the strategy is generally applicable to PBPK modelling, with only some components specific to GastroPlus. Can I have the next slide, please? So this is our publication. The full publication is available as open access in the Journal of Clinical Pharmacokinetics. And so far, I think there have been around 5,000 downloads and 5 citations in the 6 months since it was first online. If we move to the next slide, you'll see the key points from this presentation, as well as reading the manuscript. The 3 take-home messages are: 1, please join in the assessment. Try out the proposed strategy for the first time in human predictions based on the best practices and experience of the user group for yourself in your companies when making first time in human predictions. This will help us determine if PBPK model verification in preclinical species adds value to first time in human PK predictions. And the third key point is to remember that uncertainty analysis is a key consideration to obtain maximal value from first time in human predictions. If we move to the next slide, PBPK is a platform for integrating data. And we've defined these decision trees or flow diagrams that help identify the most appropriate inputs for the key pieces of what is essentially a jigsaw puzzle. As you can see from the list on the screen here, we have decision trees for metabolism elimination, distribution, and oral absorption. And we will talk you through these decision trees. Firstly, I will start off with the QSAR or QSPR plus PBPK assessment. So if we move to the next slide. It's commonly acknowledged that PBPK models require a large number of inputs. Many, however, can be predicted directly from structure using quantitative structure property relationship models. These can either be from commercial software programs such as ADMET Predictor or from bespoke internal company-specific in silico models. Now, accurate predictions from structure are unlikely. That's accurate predictions of pharmacokinetics. Indeed, they are possible, but it is unlikely. And the main utility is to identify prioritization of key experiments to build high-quality PBPK models through the use of QSAR. If there are some data available for some compounds within the chemical series that you're working, then you can potentially verify the predictions, and then you would be able to screen compounds virtually with some degree of confidence. So if we look at the flow diagram and take a lipophilic base as an example, you will see that blood to plasma ratio is a critical parameter for the prediction of tissue to plasma partition coefficients, and that enterocyte binding and precipitation in the small intestine may impact oral absorption. So parameter sensitivity on these parameters should be conducted to assess the impact on the PK predictions and thus prioritize the in vitro experiments. Would you conduct the blood to plasma ratio experiment first if you wanted to understand the tissue distribution, if that was more sensitive, say, than the in vivo precipitation predicted from structure? Now I will hand over to Neil to talk about the metabolism and elimination flow diagram.
Thank you, Neil. So prediction of clearance is obviously key to a successful first-in-human dose prediction. To achieve this, we believe that the first step is a qualitative assessment of the major route of clearance. And to do this, we recommend you consider the extended clearance classification system. Most of you will have heard of this classification system, which is based on the compound's physicochemical properties and was able to show a successful classification via the major routes of clearance. This system was coming out of work done at Pfizer, and there was also similar work done at Novartis. Okay, so to follow through the diagram, let's first start with the metabolism. So if the model is indicated as majorly cleared by metabolism, then the thing to do is to look for an in vitro, in vivo correlation for clearance. Obviously, consideration of binding is key to such correlations. Also, we recommend to consider what is the most appropriate in vitro tool. So obviously, if oxidative metabolism is predominant, then microsomes could be appropriate, whereas hepatocytes have a more full complement of enzymatic routes. So also consider the reaction phenotyping that you will be doing to characterize your molecule in animals as well as in human in vitro systems. So if you find that your in vitro data is able to predict in vivo clearance, then you can proceed with more confidence to scale from in vitro human relevant data. If not, then you may have to go back and consider, are there other extrahepatic clearance routes which should be considered? Is active transport possibly playing a role? And proceed further, see if you can understand better. If not, eventually you may have to use an empirical scaling factor based on the preclinical species, and we'll be talking about that in one of the examples we will share later. If ECCS indicates renal elimination is predominant, then we also recommend you look at the preclinical species data and compare your renal clearance in the different species with the glomerular filtration of unbound drug. If this indicates you are likely to have secretion, an active secretion process involved, then there are indeed mechanistic models which can be used, and our paper gives references to some of those publications of those mechanistic models for clearance. Biliary elimination is also quite challenging because of the role of active transport. However, again, the animal data can be helpful. In particular, monkey has been shown to be the best predictor for human biliary elimination. And also in vitro systems can be very helpful here, in particular sandwich cultured hepatocytes, and estimation of the biliary excretion index may be valuable. So taking all of this into account, then you should make your prediction, and don't forget to include that uncertainty range, which we'll be coming back to mention later. And with that, I think we're going to move on to distribution component, and I'll like to hand over to Vera for that.
Thank you, Neil. So for the distribution, for those of you who are familiar or have done any PBPK modeling, you know that there are a number of mechanistic methods predicting the tissue distribution from drug-dependent properties and from the tissue composition. Unfortunately, these are not perfect, right? They are predictive methods, and they do not account for all the possible mechanisms that might be taking place for the distribution of specific compounds, right? They are based on simple passive processes, assuming that the distribution into tissues is really driven by the passive processes and by the blood flow. So you may still, even though these are mechanistic models and for many cases do provide nice prediction, you may still have to do some troubleshooting for the distribution for your specific compounds. And this flowchart describes possible steps that you may need to take in order to troubleshoot if the distribution is not initially predicted well for your compound. So the first step, of course, you start with the default method, try to do the prediction based on the compound properties, and if you are lucky enough, it works and you are done. Right? If there is some misprediction in the animal data, and here we do recommend, I forgot to mention, we do recommend to have IV data in animals because it does really provide for a cleaner prediction of the distribution. So if the initial default prediction is not working well, the next step might be if you have quantitative whole body data available in rat. These are not very common, but in some cases you might have those available. You could use those to estimate the distribution and maybe scale or predict the human distribution, at least accounting for differences in FUP. Just a word of caution with these, make sure that if you are using this kind of data that you are looking at the correct compound, right? That the distribution is not lumping together the parent compound as well as metabolites into the same distribution or into the same KPs, because those may be different for parent compound versus metabolites. If you don't have the quantitative data and you need to do some troubleshooting, there are certain steps or certain processes that you could try. One is check the sensitivity on the 4 main input parameters into these KP prediction methods. And the 4 main parameters are listed in that first box: log P, pKa, fraction unbound in plasma, blood to plasma concentration ratio, to see which one would be the most sensitive and whether a small change in that parameter would allow you to predict the distribution in the animal species. One case that is highlighted there is adjusting blood to plasma ratio for basic compounds, which was a workaround that we were using to account for lysosomal trapping of these strong bases or molecular bases. Hopefully you will not have to keep doing this adjustment in version 9.7, but the KP method has been improved to account for it automatically based on the experimental blood to plasma ratio, but you may still have to do some exploration of the sensitivity to the parameters. If that is not a successful approach, you could take a look at the unbound volume of distribution or unbound VSS across different animal species. If it is comparable across different species, you could make an assumption that it will be similar also in human and scale it to human by accounting for differences in fraction unbound in plasma. You could also explore whether you are seeing a systematic prediction error in the preclinical species and possibly apply that prediction error also, or scaling by similar prediction error also to human prediction. And last but not least, you may have to consider using different tissue type. And this is decision where even the properties of the compound may help you to make this decision up front. If you are dealing with larger molecule or very hydrophilic molecule, you may already have a good estimate that the permeability through membranes will be low, which means the simple perfusion limited tissue model may not be the best one to account for distribution into the tissues, and maybe you should be using the permeability limited tissues, using the extracellular KP for the distribution to extracellular space, and see if you can fit the same specific PSTC across all of your animal species, and then you could use that one for extrapolation into human as well. You see at the bottom, left, right, there is one box. Also, these different strategies may still not cover all the different scenarios. If there are significant effects of transporters that are also affecting not only clearance, but also tissue distribution, or if you are dealing with possible specific binding to specific tissue components for a given compound, you may need to do some additional investigations, maybe doing some in vitro assays to identify what mechanisms might be affecting the distribution of your compound and what additional mechanisms you may need to incorporate in the model for your specific compound. And next we will move on to the oral absorption, and I'll hand this off to Aki.
Okay, thanks. So firstly, for oral absorption model evaluation, one important thing is to have IV PK data for all of the animal species you are using for the exercise. And this is to ensure that your description of distribution and elimination kinetics is accurate enough to allow focusing on really absorption related mechanisms. And in case that your predictions are not able to capture the animal PK, animal oral PK directly, then performing sensitivity analysis to determine the most sensitive inputs, whether it's related to solubility and dissolution, passive permeability, or other processes such as chemical degradation, formulation, dependent things, intestinal metabolism, and transporters. For permeability prediction, you may scale it from in vitro, for instance, KCO2 data. It's important to have the scaling done from an assay that the correlation to in vivo permeability has been established. And then in case still the permeability seems to be off, then the animal data verification to against the animal oral PK data may be used to adjust the permeability inputs and to get more accurate prediction of permeability in human. For solubility and dissolution part, it's also important to point out that the animal oral in vivo data should cover the human relevant doses and formulations to allow evaluation of formulation specific parts of the model, including dissolution and the particle sizes and so on and so forth. It's important to have a measured solubility data of the compound form you have in your formulation, and unless you're predicting based on aqueous solubility, complete and rapid dissolution of your compound, then having biorelevant solubility data to account for impact of bile salts on solubility may be important. And more lipophilic your compound is, more likely it is that biorelevant solubility is important to have. And also for basic compounds that may dissolve in acidic stomach and then precipitate after transit to higher pH small intestine, the precipitation time, precipitation rate may be optimized using the preclinical species data. And to get a reasonable predictions of precipitation rate. And in general, we try to avoid adjusting physiological parameters, but some of the GI tract physiological parameters may be variable or not accurately defined. For instance, GI tract fluid volumes may be adjusted to some extent to capture the behavior of the compound you observe and increase confidence on your predictions. In addition to solubility, dissolution related things and passive permeability, if you have chemical degradation, intestinal metabolism, or transporters, then additional quantitative data is probably required to describe those processes. And in the next slide, we have a flow diagram on intestinal metabolism. Majority of the intestinal metabolism prediction work has been done with cytochrome P450 mediated metabolism, mainly SUP3A in gut wall. And this flow diagram was initially established in SUP metabolism in mind, but in general, the flow diagram applies for other enzyme families as well. So the main point here is to try to leverage from the hepatic metabolism data you always have for your compound to evaluate, try to make assumptions that are likely to overestimate gut wall metabolism rather than underestimate. And if with those assumptions you are predicting no gut wall metabolism, then you are probably on the safe side, and there is no need to evaluate or spend more effort on predicting gut wall metabolism. So firstly, the question is that is the compound metabolized by enzymes that are present in enzymes or enzyme families that are present in the gut wall? For instance, cytochrome P450s are metabolizing your compound, then it's possible that SUB3A is metabolizing your compound in the gut wall, and it's relevant to ask the question that if gut wall metabolism plays a role. Then you may take the liver metabolism data and make the assumption that all of the metabolism in the human liver microsomes or hepatocytes is via the highest abundant enzyme in the gut wall. So in the case of P450s, SUB3A. And use the known abundance of the enzyme both in liver and gut wall to scale metabolism to gut wall, and evaluate whether that gut wall extraction is significant or not. If it is, then reaction phenotyping data to identify that which enzymes are relevant. You can exclude the enzymes that are not present in the gut wall. That may be useful, and you would still probably have intrinsic clearance data. And you could use that for predicting gut wall metabolism. But knowing that concentrations in the gut wall tend to be relatively high, and saturation of gut wall metabolism is possible. So then if you are still predicting considerable gut wall metabolism, then having Vmax and Km for your metabolic reaction may be useful for more accurate gut wall metabolism predictions. And obviously this flow diagram also goes to the possibility that none of these are possible to do based on liver metabolism data, and then you should consider measuring metabolism in intestinal in vitro model and use that for capturing gut wall metabolism. And then on the next slide, Neil Parrott will continue on uncertainty and variability.
Yes, thank you, Aki. Obviously, the default mode in GastroPlus is to deliver a single average profile. However, if you do this for your first in-human prediction, we believe you're severely limiting the value of PBPK, and it is not recommended to do that. What we would recommend is that you perform an uncertainty analysis and that you convey the results of that analysis with your first in-human PK prediction. We recommend that the way to do this could be using a parameter sensitivity analysis. Parameter sensitivity analysis is well recommended for PBPK modeling, and in particular, recent health authority guidance documents specify the use of parameter sensitivity analysis. But this is particularly valuable for first in human, because if you can identify the most uncertain modern parameters and then vary those within a realistic range, you can then convey the uncertainty in your simulated PK profiles. And this can be very important to do when planning for your first in-human study. So please do consider this and convey uncertainty whenever you deliver your first in-human dose recommendation. With that, I think we should move on, and so I'm going to hand over again to Neil Miller.
Thank you, Neil. So now we're going to move from theory to practice, and we're going to describe 4 case studies that focus on more challenging compounds to illustrate and highlight key components of the strategy of the diagrams that we've presented. Specifically, we're going to look at empirical PBPK model factors. We're going to look at the impact of blood to plasma ratio in predicting the volume of distribution for a basic compound. We're going to talk about lipophilic weak acid and the challenges that presents with the low solubility for in vitro assays. And finally, we'll look at another case study which will highlight using a permeability limited PBPK model. So first off, it is Neil to talk about the empirical PBPK model factors. Thank you, Neil.
Thank you. So if we could have the next slide, please. Okay, great. So in choosing the case studies for this paper, we wanted to choose compounds which were particularly challenging in order to illustrate that PBPK modeling can be valuable even for such cases. This example is a very highly lipophilic neutral molecule, and it has very challenging properties for in vitro measurements. In particular, the fraction unbound in plasma is so low as to be hardly quantifiable. Clearance was mainly by metabolism, but again, with that high log P, the aqueous buffer solubility was really below the limits of our assay. And although we had biorelevant solubility data, which was of a higher order, and that was used in the modeling. The permeability was estimated to be reasonable. So the first challenge with this and where we used an empirical approach was in the scaling of clearance. Obviously, with such high binding to proteins, then it was not possible to take account of free fractions in vitro and in vivo in the scaling. What we did was to develop a method of in vitro to in vivo scaling in the rat, whereby an empirical scaling factor could be estimated. This was then verified in other preclinical species where we had IV data, namely dog and monkey. And as it worked well to scale from in vitro hepatocyte data in those species, we felt more confident in scaling up from the human data. This nonetheless delivered a very low human clearance expected of around 0.5 milliliters per minute per kilo. We also needed to do some empirical modeling for the absorption because that was particularly challenging. However, we had some dog data where different formulations had been dosed in the dog, and so we optimized the absorption model, the GastroPlus ACAT model in the dog, and in order to match for those formulations, and then we applied those optimizations for the human simulation. The optimizations involved adjustments to the percentage water in the intestine and adjustments of particle size for the different formulations. On the right-hand side of this slide, you see the simulated profile, which, as you can see, was well covering the Cmax of the first-in-human dose data in 6 healthy volunteers, also covered well the range of exposures. However, if you look at the little inset, you will see that this compound had an extremely long half-life, and using the average predicted clearance of 0.5, we quite significantly underestimated that half-life. However, if we had taken the uncertainty range around the clearance, which was 0.2 to 0.7, then we would have just about picked up those last sampling points in the human study. So again, really illustrating the importance of taking account of uncertainties in the data and conveying that when you deliver your prediction. And with that, I'd like to hand over to Neil, I believe, for case study 2.
Thank you again, Neil. So this case study also looks at a lipophilic compound with a lipophilicity of 4.2, and it's basic in nature. So typically for a compound like this, there's a challenge to predict the volume of distribution. And this case study will highlight the power of the blood to plasma ratio in predicting the volume of distribution of bases, because it's used to estimate the very influential parameter of the association constant with acidic phospholipids. So if you look at the table on the left-hand side, you can see there's species differences in that blood to plasma ratio, with rats, rabbits and dogs having a ratio greater than 1, so there's more association with the blood. And mini pig and humans, the blood to plasma ratio is less than 1, so there's more association with the plasma. For this example, we're focusing on IV data. So we use the non-compartmental analysis clearance alongside a complete set of compound-specific inputs for predicting the tissue partitioning. So as Viera says, that's log P, the lipophilicity, pKa, plasma protein binding, and blood to plasma ratio. This allows then a retrospective isolation and assessment of the blood to plasma ratio on predicting the volume of distribution, because there isn't any uncertainty in any other parameters because they've all been measured. So if you look on the table on the right hand side, you can see that the Lukacheva KP method predicts the volume of distribution within 60%, so that's a lot better than twofold for all species except the monkey. This does highlight quite nicely the weight of evidence approach that's used in discovery. So here, when you can see 4 out of the 5 species are accurately predicted, then that method would be considered verified for use in predicting for human. Obviously, this example has multiple species, and you may be working with a compound where data is only available in a smaller number of species, and then any outlier would probably need to be investigated further. It's worth noting that the measured blood to plasma ratio is critical in predicting the volume of distribution for this basic compound. But what I should also point out is we never looked into the plausibility of markedly different species-specific association constants with acidic phospholipids, because if you've got a different blood to plasma ratio and the plasma protein binding is fairly consistent across the species apart from monkey, you will then back calculate very different association constants with acidic phospholipids. That wasn't looked into, but this example very nicely highlights the need to measure the blood to plasma ratio when predicting the volume of distribution for a basic compound. So with that, I'm going to hand over to Aki to talk about the next case study.
Okay, thanks. Yeah, now we have the slide. Yeah, so here we have a lipophilic weak acid with low solubility, and yeah, with this also the challenges include the fact that the characteristics of this compound are beyond the limits of in vitro methods. So fraction on pound in plasma is below 0.1%, something that you are not able to measure accurately. And also permeability is something that could not be reliably measured. Clearance could be measured in vitro, but due to the fact that the compound is highly bound in plasma and free fraction in plasma is not known, clearance prediction is also very uncertain, and the free fraction in plasma also uncertainty in free fraction in plasma propagates to uncertainty in volume of distribution predictions as well. First, on the solubility and permeability part, enabling formulations increased solubility quite significantly, and in vivo preclinical data suggested good absorption at clinically relevant doses. And with that, absorption was not considered as a key source of uncertainty in first-in-human predictions, but volume of distribution and clearance were quite uncertain. And then the in vivo IV data on dog and monkey suggested low clearance, low volume of distribution, whereas rats showed relatively high clearance and high volume of distribution. And a plausible explanation for this kind of behavior is species differences in fraction on pound in plasma, and parameter sensitivity analysis around that suggested that plausible range for fraction on pound in plasma is from 0.01% to 0.1%, and that also provided a plausible range for simulating scenarios in human low clearance, low volume scenario, and high clearance, high volume scenario as well. And that was used as a kind of uncertainty range for first-in-human predictions, and eventually the observed data in human fell quite nicely within that uncertainty range. So the take-home message with this is that in vivo preclinical evaluation and verification of the PBPK model allowed identifying the key source of uncertainty and determining a plausible range for that uncertain parameter to generate reasonable and useful scenarios for first-in-human predictions. And then I hand over to Vera for the next example.
So in the final example, when the slide gets there, we'll discuss again more of the issues of distribution. And this is the more complex case where we needed to employ permeability limited tissue model. This was a compound which had high molecular weight and large number of polar potentially ionizable groups. So based on the properties of the compound, we already did have an indication that this compound may require the permeability limited tissues, as based on the structure and properties, it may have trouble crossing the membranes by simple passive diffusion. Nevertheless, we did try both types of models. We've tried the perfusion limited tissues as well as permeability limited tissue models. And the initial investigation was done based on the data after IV administration in rat. And if we were looking only at plasma concentrations, both tissue types actually would be able to predict the plasma concentrations in the rat. But in this case, we also had information on the concentrations in the kidney. And this is where the simpler perfusion limited tissue model was failing. In order to match both the concentration in the plasma as well as in the kidney in rat, we did need to employ the permeability limited tissues. And we ended up fitting the specific PSTC value, which then with the physiology, with the tissue volumes, was used to scale the or predict the PSTC, the permeability surface area product for every single tissue. And that allowed us to really match both the plasma as well as tissue concentration in the rat. So assuming that the membrane composition remains similar and that the specific PSTC does translate across species, we then use the same approach to predict the PK in human. In the plots, in the top right corner, you see the human predictions, and the black line and black points are showing the plasma concentration. And you can see that even the first plot with the perfusion limited tissues, you would see that it wasn't terrible. Even as for first in human prediction, even with the perfusion limited tissues, it would not be terrible. But the tissue concentration, the dashed green line would be very different predicted between the perfusion and permeability limited tissues. And again, based on the rat data where the accumulation in the kidney tissues was observed with the experimental values, we would look or use the permeability limited tissue model to predict the PK also in human. The other piece of evidence here was that really was pointing out more towards the permeability limited tissues, was or that confirmed that the permeability limited tissues might have been the better choice, was comparing the prediction with the human data in terms of the renal secretion. The all perfusion limited tissue model would predict that all of the compound is secreted in urine within about 24 hours after the administration. But the observed data or the clinical data were showing that the renal secretion was actually lower within that time frame, that it took longer. So, and the model with the permeability limited tissues really allowed better to match not only the plasma concentrations in human, but also the kinetics of the renal secretion and the amount that actually got excreted within that 24-hour period. And that's the end of the examples. And I believe Neil has the final slide.
Thank you, Vera. So this brings us back to the key points I mentioned at the start of the webinar, in that we would like you to help us by joining in the assessment. Try out the proposed strategy and the flow diagrams for your predictions. And by doing that, you'll help determine if or if there is real value in the PBPK model verification in preclinical species, because as I said at the start, I think that's subject to scientific debate at this point in time. And as Neil stressed with his flow diagram, you must remember to incorporate uncertainty analysis when you're making these predictions. There's things you'll learn when you're moving through the preclinical data. For example, around clearance, it may be there's no in vitro in vivo correlation. You may have a range of clearance with respect to liver blood flow in the preclinical species, and that should be factored in to your final human predictions prior to the first time in human study. With that, I would like to thank you for your time and attention. I'd also like to mention the efforts of Michaela Reddy in helping us get this manuscript out there. Unfortunately, Michaela couldn't be with us today, but she was very instrumental in driving this manuscript forward. And now it is your opportunity to ask us any questions. Thank you.
Great. Thank you all very much to all the speakers for the great presentation. At this point, we will open up for questions. Okay, I see the questions coming in here. First question I'll read out loud. Could you briefly mention any specific considerations for irreversible covalent inhibitors in which blood plasma stability and target engagement may be an extrahepatic route of clearance? It's a long question. Hopefully you can hear that clearly.
Yes, I mean, I can start the response to that question. It sounds like it's a very complicated compound, and on one of the flow diagrams, I think Aki pointed out that if you have more complicated compounds where more mechanisms are at play, you will need more data to define those mechanisms. So for compounds like that, you would need additional data to accurately predict using PBPK.
Okay, thank you. Here's the next question. In comparing the sensitivity of different predictors, how would you rank different factors, solubility, permeability, log P, BPR, etc.?
Yes, so in the uncertainty analysis, we recommended using PSA, parameter sensitivity analysis. Obviously, you limit it to a maximum of 2 varying at one time. You can do that in GastroPlus using a 3D plot, but if you want to consider multiple factors, then you will have to do that one by one, and then maybe consider combinations if you think they are particularly likely to occur. Then that would give you maybe the extremes in your expectation.
I would just add.
I think example. Sorry, Vera.
Yeah, I just wanted to add that, you know, this is really going to be dependent on the compound. So looking at the compound properties and what type of compound you are dealing with, you'd probably a priori be able to already, you know, make a decision which parameters are likely to have bigger impact. That.
All right, thank you. The next question: how to determine those for first-in-human PBPK model?
Yes, so we didn't mention this. We were concentrating on PK, but obviously we will all be linking this predicted PK to some kind of PD data, ideally using PK/PD modeling. Of course, you have the capability to do that very conveniently using the PDPlus module. But in any case, some kind of link to target occupancy or expected efficacy in some preclinical models should really be brought into play in order to select the dose, as well as safety, of course. But that was not really part of our analysis, although it is just as important as PK prediction.
Thank you. Here's the next question. Can you briefly summarize the benefits of using PBPK for first-in-human estimation over simpler empirical approaches? Also, this is the first question. Also, is the complexity of these models appreciated by authorities with regards to a better understanding of your compound? There's essentially two questions.
So I think the advantages from my point of view of PBPK over empirical predictions is that you're using all of the data. Certainly when you're using allometry, you'll focus more on the parameters and not the pieces of the jigsaw puzzle that we've talked about. So if you look at distribution, the lipophilicity, the pKa's, the plasma protein binding, the blood to plasma ratio, that would be ignored if you're allometrically predicting volume. So you're using everything you know, you're combining that with physiology. And we're not saying that the predictions are going to be accurate every time. We're saying that there will be uncertainty in that and you should take that through. But I think the other value of the PBPK modeling is when you put the pieces of the jigsaw puzzle together and they don't fit, you know then the risks in your predictions when you go to first time in human. With allometry and that extrapolation, sometimes you're unaware of the risks. So I think you're more informed about the potential to get an accurate prediction by combining all the data and integrating that with physiology within a PBPK model. And I think in terms of the second question and the regulatory expectations are that PBPK models will be in submissions now, as we've seen lots of guidelines coming out from the FDA and the EMA. So the regulators are expecting it, and I think there's even examples where if you haven't done PBPK modeling and you have supplied the parameters to build a PBPK model, the regulators will consider building the model to assess your compound.
Thank you. Here's the next question. How to handle nonlinearity PK, especially when nonlinearity extends differ across different species?
Okay, so in terms of the clearance which I was describing, of course, it is possible to measure in vitro clearance at a range of concentrations with human hepatocyte studies, for example, in order to characterize expected nonlinearity in hepatic clearance. I would be cautious about extrapolating nonlinearity from animal species, but it may also be raising a flag that you need to look out for that. Particularly as Aki mentioned in his slide, saturation of intestinal metabolism is quite common.
Yeah, and maybe add on that if you don't have in vitro data that would suggest nonlinearity, then the PBPK model may be used, parameter sensitivity analysis may be used to evaluate that what mechanism could be saturable and explain the observations you have in vivo. So that could direct your experimental work, that whether you should look at saturation of metabolism or transporter effects or precipitation of the compound in the intestine. So parameter sensitivity analysis may guide your experimental efforts.
Thank you. Here's the next question. How do we handle renal transporters which are not expressed in rodent and animal models, such as OAP3?
Yes, in my slide on clearance, you may have remembered that I mentioned there are some in vitro models, and in our publication you can find the references to those models. So human kidney cell line expressing OATs has been explored, and a quite reasonable in vitro to in vivo correlation was shown for a set of molecules. So that would certainly be worth looking at.
All right, thank you. The next question. How factors such as biophysiological aspects of aging from infant to old age affects the PK/PD of drugs? At the same time, how stress-related changes in blood and gut inflammation caused by food and microbials affects PK?
Maybe I can just comment on the first part of that question, which is about predictions in different age ranges. Obviously, PBPK is the recommended way for predicting in children and the very young, because you have the potential to account for the immaturity of physiological processes using PBPK. In particular, there's quite some growing knowledge on the differences in expression of enzymes in the young. So that would be the recommendation. But usually the first step is to verify your PBPK model with adult data.
To follow up on that question, so yes, for the PK in the children, and you can see it in the literature, there have been a number of successful cases already presented on scaling accounting for the physiological changes. Of course, looking at the PD is a different story. Right? The key part for that one would be making sure that you have well characterized the dose-response relationship, and of course then you would be also assuming that the disease is the same in children as in adults when trying to do that scaling. Right? So for PK, we are making a lot of good progress for predicting PK in children. For PD, there is more uncertainty. For the second part of the how stress-related changes are affecting PK, the bigger question, or the first question that we would need to get an answer for is how the stress changes the physiology. Once we know what physiological changes are happening in different conditions, then the PBPK models are the ideal platform for actually incorporating those physiological changes. But for many conditions, this relationship between the condition and the actual physiological change is not known.
Thank you. Okay, next question. Are there any 3D printing tissue models available to generate the data which can be correlated with GastroPlus?
Well, that's an interesting question. There are, of course, new models emerging, so-called organ-on-chip models. There's some publications of quite exciting new developments in this area, although I have not seen any strong, I would say, strong validation of those for in vitro to in vivo scaling. There is some limited verification done. There was a nice paper from Samandouros et al., I think a couple of years ago, but it was for a very, very limited number of molecules. So I think we still need to see more data from those systems before we can start to use them for our routine work.
Well, thank you for that comment. I guess we're reaching to the end of our time, and we still have a few more questions that cannot be answered because the time limit. But I do suggest that for this. You can go to the GastroPlus User Group LinkedIn website where you can post your questions. I think we could probably, from our side, we can collect those questions afterwards, we can post these questions in our website, and then we'll address those questions as much as possible. So now there is another poll here, and hopefully you can quickly answer it. And now I want to thank you again to our speakers for your presentation and answering these questions. For the audience, thank you for all for your participation. You may now disconnect from the webinar. Thank you, everyone.